Fermentation is a natural metabolic process that occurs in the absence of oxygen, where microorganisms such as yeast or bacteria convert carbohydrates, such as sugars or starches, into other compounds such as alcohol, acids, or gases. Here's a simplified explanation of how fermentation works. Yeast activation. In the context of alcoholic fermentation, yeast is the microorganism primarily responsible for converting sugars into alcohol and carbon dioxide. Yeast cells are activated when they come into contact with a sugar-rich solution, such as grape juice in winemaking or wort in beer brewing. Sugar breakdown. Once activated, yeast cells begin to metabolize the sugars present in the solution. Yeast enzymes break down complex sugars such as glucose and fructose into simpler molecules such as pyruvate. Anaerobic respiration. In the absence of oxygen, yeast cells undergo anaerobic respiration, a process where pyruvate is converted into ethanol, alcohol, and carbon dioxide. This process releases energy that the yeast cells use to grow and reproduce. Alcohol production during fermentation. Ethanol accumulates in the solution as a byproduct of yeast metabolism. The concentration of alcohol increases over time as more sugars are consumed and converted into ethanol. Carbon dioxide release. Along with ethanol, carbon dioxide gas is also produced during fermentation. This gas is responsible for the bubbling and foaming that often occurs during fermentation. In some cases, such as in bread baking, the carbon dioxide produced by yeast helps leaven the dough, causing it to rise. Temperature and pH control. Fermentation is sensitive to factors such as temperature and pH. Different strains of yeast thrive in specific temperature ranges and pH levels, so maintaining optimal conditions is essential for efficient fermentation. Too high or too low temperatures, as well as extreme pH levels, can inhibit yeast activity and affect the fermentation process. End of fermentation. Fermentation continues until either all available sugars are consumed or the yeast cells reach their alcohol tolerance limit, at which point they become dormant. The fermentation process can be stopped intentionally by cooling the solution or adding chemical agents, or it may naturally cease once fermentation is complete. Overall, fermentation is a fundamental biological process used in various applications, including food and beverage production, e.g. wine, beer, bread, yogurt, as well as in industrial processes such as biofuel production. It allows for the conversion of carbohydrates into other useful compounds under controlled conditions, providing a source of energy and creating a variety of products with distinct flavors, textures, and properties.